Happy to introduce Jenny Gallop from Cambridge University. She's actually a Wellcome Trust Senior Research Fellow at the Gurdon Institute and an Associate Professor in the Department of Biochemistry. And she has an interest in the regulation of endocytosis and actin, focusing on the proximal tubule. And this will be a combined presentation. Jenny will start, and then I think there will also be a contribution by Matias Simons, who is my co-organizer of this workshop, and he's a professor of human genetics at Heidelberg University, and also focuses his current research on proximal tubular receptor protein reabsorption. So now we move to Dent disease and Lowe syndrome.
Okay.
Jenny.
Yeah. Well, thank you very much for the kind introduction, and thank you for accepting a cell biologist into your group. Okay, failing at the first. Do I point it?
The arrows?
Yeah, I did that as well.
Yeah.
Is it the Zoom thing?
Should. Oh, actually do. No.
Okay.
No.
Okay. Thank you. So what I'm trying to do in my lab is to understand the molecular logic underlying actin regulation. And this problem exists because the actin monomer is, a soluble monomeric protein in solution. And to actually become into the actin cytoskeleton, the structure of filaments, that's the active process here, that's also the subject of regulation at many different steps. So I'm showing here the filamentous forms. You have bundled actin filaments. You can have branched actin filaments, for example, with the ARP2/3 complex, which are the relevant ones that I'm going to be talking about later. You have cofilin linked actin filaments, which helps these actin filaments disassemble. So the actin cytoskeleton, this assembly into these different structures, is controlled at the level of signaling through Rho-type GTPases, protein kinases, by transmembrane receptors at the membrane surface, and also by a lipid metabolism. And I'm trying to understand how these networks of positive and negative regulation impact on the actin cytoskeleton, and so that we can learn how to change them. So the underlying question that we're asking at the moment is, can we understand and harness the specificity that the cell uses? Because obviously, although actin is required all over the place in all different types of cells, somehow the cell is tuning that actin arrangement in the right place where it needs to be at the right time. So if we understand that biology, can we then harness that specificity to target aberrant actin in disease? So I've become interested in Dent disease and Lowe syndrome. So Dent disease patients present with hypercalciuria and other renal complications, of which you all know more than me. And low molecular weight proteinuria is a particular hallmark. There's the onset in childhood with worsening renal tubular dysfunction and end-stage renal disease by the ages of approximately 30 to 50 years old. There's two genes particularly associated with Dent disease, and also some Dent disease that's unexplained. So Dent1 is due to mutations in CLCN5, which is a chloride transporter, and Dent2 is caused by mutations in OCRL, and this is a lipid phosphoinositide 5-phosphatase, and that's how I've become interested in this, because phosphoinositide 4,5-bisphosphate is a key regulator of the actin cytoskeleton. So likely the incidence of both Dent disease 2 and Lowe syndrome, which is also caused by mutations in OCRL, are around 1 in 500,000. And Lowe syndrome is a multi-system disorder that's more severe than Dent 2, typically diagnosed first by the congenital cataracts, but also other effects, hypertonia and intellectual disability and other problems. So at the cellular level, the loss of this enzyme leads to aberrant actin polymerization. So this is the process, the hydrolysis of the phosphate group by OCRL here on PIP2, and this is normally enriched in the plasma membrane. It's a major signaling phosphoinositide of the plasma membrane. And in healthy cells, what we think is happening is that the PIP2 is helping to assemble the clathrin-coated vesicles and other things, which causes a transient burst of actin that leads to that trafficking of the vesicles into the cell, and then the further endosomal pathway as these proteins are taken up. And there's a stepwise phosphoinositide conversion into other lipids, in particular PI3P, that's enriched at the early endosome. So in the patient cells, the OCRL has got a sequence in it that interacts with the clathrin machinery. And in this case, there's an elevated level of PIP2. The PIP2 can't be degraded as there's progression through this endosomal pathway, and so there's elevated levels of PIP2. This leads to increased actin polymerization and aberrant actin aggregates around the endosomes and the cells, and particularly some, as was shown by nice work from Antonella de Matias, this leads to a block in the endosomal trafficking here. You can see the megalin receptor uptake is decreased here, characterized by these basket structures around the endosome. And importantly, she showed in 2010 that if you target those actin assemblies, then that can lead to re-increases in that receptor internalization. So I took on board those findings and fitted them in to our own developing picture of the further regulation that we were characterizing as happening of the actin cytoskeleton. And this is a complicated picture here, which is really to illustrate that there's a network effect within the phosphoinositide and also in the actin regulation. I'm not showing the details here, this is very much an overview. But what I did in this picture was basically add in this arrow here that the three phosphoinositides were working together with PIP2 in order to impact on this actin machinery. And so the thought was that if we use alpelisib, which is a newly developed, very specific class 1 PI3 kinase inhibitor developed for cancer, that we could dampen down this 3-phosphoinositide sort of pathway that's impacting on the actin machinery and compensate for the elevations in PIP2. So we show in vitro that the PI3 kinase inhibitors could reduce those F-actin assemblies, and this was indirectly, not directly to do with OCRR, but indirectly via a network effect. So we started a collaboration with Olivier de Voislab and particularly Marine Burquez, who's here in the audience. And these are her images from primary proximal tubule cells from the mouse model with an OCRL knockout. And you can see here in the normal proximal tubule cells, looking at the endosomes here in pink, with the yellow actin assemblies around them, you can see this normally happens to some extent. And then with the OCRL knockout, you can see the actin baskets that end up surrounding the endosomes. And with treatment of these cells with alpelisib, then you decrease these actin assemblies, as predicted from our work in immortalized cells and the extract systems. So this work was published in 2020, so I'm just showing you some of the highlights here of this work with the reference on the slide. So Marine treated these model mice in vivo, showing that the alpelisib improved the proteinuria. So this is the phenotype of the mouse. It's a very good model for the human disease. It's a humanized model with a knockout of INPP5, and then the of the mouse isoform, and then the re-expression of the human INPP5B, and that's because of the clathrin site within the mouse isoform. And so they have elevated low molecular weight proteinuria and albuminuria. And we saw around a 35% improvement in CC16 excretion and albumin excretion after a 6-week treatment with alpelisib according to the standard protocols that were used in mice. So taking then cross-sections through the proximal tubules of the mice that have been injected with the labeled lactoglobulin, you can see the decrease in the absorbed protein in the proximal tubules here in the knockout mouse model, and then this regaining the protein in the proximal tubules with alpelisib treatment. And is accompanied by an increase in the levels of the megalin receptor, which usually becomes degraded likely because of the block in the endosomal trafficking. So this is the overall model here, that usually what happens is that an endocytosis of the megalin receptor in the proximal tubule that's responsible for reabsorbing those proteins that are being filtered out by the glomerulus. There's usually this activation of class 1 PI3 kinase alongside the PIP2, leading to a short burst of actin polymerization that then allows this continued absorption and the endosomal trafficking here. when you have a lack of OCRL, then there's a sustained increase in actin polymerization. These actin aggregates surround these endosomal compartments. You're no longer able to recycle the megalin receptor back up to the membrane, and instead it becomes degraded. But with alpelisib treatment, although there's still the elevated levels of PIP2, we can rebalance that actin regulation and sort of resume this trafficking pathway. And we can also show there are also knock-on effects by having these actin aggregates that we get improvements in the rest of the actin architecture of the cell. And we think that's likely because the other components are then being able to relocalize back to where they should be working instead of being clustered around the endosomes. So in summary, we found that alpelisib normalizes the key disease phenotypes in the immortalized cells, in the primary proximal tubule cells, and also within the humanized mouse model. So alpelisib is an already approved drug. It's marketed as Piqray for PI3 kinase positive breast cancer, and it's also this year been FDA approved in the rare disease situation for chronic use in PI3 kinase related overgrowth syndrome. So it has a moderate profile of side effects. Hyperglycemia is a major on-target side effect, and that's because it's a direct inhibitor of class 1 PI3 kinase, so that's expected and inevitable. In preclinical studies, alpelisib is not supposed to be crossing the blood-brain barrier, but in clinical use, there are some indications that it does. For example, some of the PROS children that suffer from seizures have seen an improvement when they've been on alpelisib treatment. So we opted not to file a second medical use patent, really to not get in the way that it was inevitably going to be owned by Novartis. And we considered whether to attempt off-label development and considered that patient access would be limited if we went for that route. So we've been embarking on discussions with Novartis about an investigator-initiated trial. We're looking ahead. I'm part of the ERKNet consortium that's been introduced, looking ahead to an innovative trial design for alpelisib in Dent disease 2, and this is with Francesco and here's the major work that he's done. So our thoughts are that Dent disease 2 is a good start point for a proof of concept of alpelisib therapy in the renal tubular dysfunction caused by OCRL deficiency. And that's because we really have no data on anything to do with the neurological dysfunction. The mouse model is not a good not a good model in that respect. Also, the Dent 2 patients before the onset of chronic kidney disease can be young adults, and they don't have intellectual impairment. So our dosing, the idea is that we'd start low and then increase to the level established for PROS that's now being used on a kind of chronic basis. We're looking at doing a pilot study in a small number of patients for a short time to establish if the safety of this drug is similar to the populations in which it's been used so far. And then if it's safe with some indication of efficacy, then we look ahead to a transition to children suffering from low syndrome. So for endpoints in the trial, we're looking at the urine measurements, low molecular weight protein, and other measurements also in plasma. And then we think important in this low patient number situation is that have a DMSA scan, a quantitative measure that looks at the protein retention in the kidney, meaning that's a very sensitive measure here. And in this disease, it's a very characteristic pattern that the radionucleotide goes into the bladder rather than being present in the kidney. And of course, this is primarily a safety trial in the first instance, looking for adverse event monitoring. So the next steps are the outcome of the funding applications, as we've heard so far, and also continuing to engage with Novartis, looking at the pathway to patient access if we were actually successful. As a basic scientist, I'm now embarking on patient engagement projects, which is very exciting for us. Our institute has had a long-standing public engagement program, and patient involvement is now an exciting new venture for the institute. And I'm looking ahead to the evidence that's needed for low syndrome. So in my studies looking at actin regulation, I work a lot with Philopodia. So this is a growth cone system here, Philopodia actin-rich structures. So we're looking ahead at what evidence we can gather to justify a transition into low syndrome, and what preclinical neuronal models are going to be worthwhile. So interestingly, PI3 kinase beta inhibitors have been repurposed for Fragile X syndrome. So there are some assays and things that are established that we could work with. And thank you very much. I'd like to particularly acknowledge Olivier Devoislav, because who's here, Francesco Emma, who's working with, we're working with on the trial, and John Gatsby, particularly in my lab, who's been working on this project. Thank you.
Yes, so we'll just continue with Matias's talk and then have a joint discussion.
Okay, so hello everyone, and I'm gonna try to fit a small unpublished story that we have in the lab in the next 10 minutes before the lunch break, and this is a story that is part of the ERKNet consortium also, and may or may not be related to what Jenny just talked about earlier. So Jenny did a good job in describing the Dent disease, and I don't have to say much. Just to repeat that we're dealing with two types. One is caused by CLCN5 mutation, which is the chloride proton exchange sitting in early endosomes. And then the other one is the OCRL1. The hallmark of this disease, as Jenny said, is the low molecular weight proteinuria, and this is mostly caused by the lack of receptors like megalin and tubulin on the proximal tubular surface. And these two diseases have a common endpoint, proximal tubular defect, but mechanistically they're not thought to be so related.
Sorry. So, yeah, okay.
But I would like, can you put the presentation? Thank you. What I would like to add though is that in both diseases, there are reports, quite a number of reports that also say that there's a glomerular involvement, not just the proximal tubular involvement. This is not clear if this is a secondary effect, so it's a consequence of the tubular interstitial inflammation, but here there's histological defects such as FFPN, and also in some cases a very high-grade proteinuria. This is now data from Marine Berquet again, who is here in the audience. She's looked at the mouse, the knockout mouse for CLCN5, and she can see indeed that there is fibrotic material around the glomeruli, and this is quantified here. Okay, so we have been looking into this by using the flies. You may wonder why you can use flies for something like this, but indeed there's a cell type called the Drosophila nephrocyte, and this is a quite peculiar cell type that exists in two populations. We mainly work with this population here. These are round cells that cluster around the esophagus, and they are actually hybrids of podocytes and proximal tubular cells. So they have both slit diaphragms here at the outer surface for the filtration of the hemolymph, and then they have the protein uptake machinery consisting of tubulin and amnionis. So they're basically presenting both features of the proximal nephron. And when looking into the CLCs, you have to consider that in humans there are 9 of them, but in flies there's only 3. So the question was which one of them is really the CLCN5. There was some data from the phylogenetic analysis suggesting that CLCC is more close than the others to CLC5. But we had to confirm this, and we started to first look at the tubulin expression on the nephrocytes as the main hallmark of this disease. And here you can see the control set. Tubulin is on the outside here on the surface. And only when we knock down CLCC, we see a loss of the tubulin. And this has the consequence that we have a reduced albumin uptake. So here is an experiment ex vivo where we basically use the endocytic tracer albumin tricks to see how the uptake works. And you can see only the CLCC knockdown has a decreased or even completely missing uptake. And You can also appreciate that the cell size is much more increased. This is quantified here, possibly as a consequence of the decreased endocytosis. We also looked at the endocytic or endolysosomal organization. Here is rabinosin 5, which is an early endosomal marker, which is very cortical in these cells, and then there's RAB7, which you cannot see so well here, but in any case, the knockdown of COCC really disturbs the organization of early endosomes, but also late endosomes. And if we look at electron microscopy, you can see that in the COC knockdown, there's really a lack of the endocytic pathway. You usually have these large early endosomes, and these are missing. But we also see these accumulations of lipid-rich materials, and we could, I'm not showing you this now, but we could see that these RAB7 clusters here are full of cholesterol. What we also noticed in the EM studies is that the slit diaphragms are actually a bit strange. So normally you only have one of them here at the outer surface, but here in the COCC knockdown, we see many of them lining the invaginations here. So there are ectopic slit diaphragms. And we looked at this in IF also, and we could see it's mostly, I think, best viewed here. So here, the slit diaphragm marker KIRA, which is the ortholog of NEF1, is not here on the surface as a straight line, but extends into the interior of the cell along these invaginations. So we can say that the knockdown of COCC and nephrocytes is a good model for Dent's disease because we, on one hand, have a decrease in the tubulin-dependent protein uptake, and on the other hand, we also have a problem with the slit diaphragms, which may relate to the glomerular defects that we see in the patient and the mice. Now, we just heard about actin from Jenny, and we also looked at this thinking that these two diseases may, after all, be related mechanistically. And normally, when you're staying with phalloidin, which labels filamentous actin, you cannot see much. There's a slight enrichment at the cortex here of these nephrocytes, but then when you knock down COCC, this is what you get. A massive actin accumulation at the cortex, also surrounding some vesicles, which we don't exactly know what they are, but this is a massive accumulation, a strong phenotype. And this reminded us, of course, of the publication from Jenny and Marine and Olivier, but also there was a paper from the Gugino lab a long time ago, this is almost 20 years ago, where they could show that COT5 interacts with cofilin. So cofilin was actually introduced by Jenny on the first slide. This is an actin depolymerizing factor, a very prominent one. And we thought this is interesting, and we started to look into cofilin. And just to give you a little background, so cofilin is an actin depolymerizing factor, as I said. It destroys actin filaments, and it can be inhibited by this kinase called limb kinase. So this phosphorylation here is making cofilin inactive. And we could model this in flies by introducing this serine in an unphosphorylatable form, and also by knocking down limb kinase. Both treatments would lead to an activation of cofilin, and we asked whether or not this would rescue our phenotype. And this is what we get. So both these conditions, TSR is the Doppler name for cofilin. This is a constitutive active form where you have this constitutive sorry, this is the inactive form. This is form. So the phosphorylated form, you can see that there's this is the active form. I always get confused with this. So the constitutive active form, you can see that you can rescue the active accumulation phenotype that you get with the COCC knockdown. And also when you knock down the limb kinase, you get the same effect. And what happens now when you look at cubulin? Indeed, in both cases, you restore the cubulin expression. And also, when you look at the albumin uptake, this is also partially, at least partially, rescued. So to summarize, the COCC knockdown, which is the homolog of COC5, leads to a strong cortical actin accumulation. And by activating cofilin function, the actin depolymerizing factor, this can normalize the actin at the cortex and partially rescue the cubulin expression and the albumin uptake. So this suggests that the mechanisms for densities 1 and 2 are maybe after all related. And this could also suggest that treatments that you have for the densities 2, like the alpelisib, could potentially also work in density 1. So the last final question is, what about mammalian COC5? I showed you a picture from the COC5 knockout mouse in the beginning, and we have an ongoing collaboration with the group of Olivier, in particular Marine, to study in how far we can reproduce these phenotypes in the knockout mouse. And I'm just showing you one result to show that also in these mice, you can see an increase in actin in the glomeruli, potentially in the podocytes, and this is quantified here. We also have some data from isolated proximal tubular cells suggesting that you have an increased actin also in those. So this is all I want to show you. This is the lab, and there are some other former members from the group that were involved in this. Now Salomon in the group who's here working on this, and we have this very nice collaboration with Marine and Olivier. Thank you very much.
Thank you. Any questions or comments?
You can, Jenny, want to come?
Maybe you can explain a little bit what are the expected side effects of the alpha.
Yeah. So hyperglycemia is the main one. The other things, I think, would be some mucosal effects, mouth ulcers. There's gastrointestinal upset. At the levels of the treatment used in PROS-CLO syndrome, these are kind of minimized. It's more at the higher levels that are used for breast cancer.
Mm-hmm. And then somebody's
so I should say that diabetic, so that would be an absolute no.
Increase of serum creatinine could also occur, somebody says here.
I'd have to pass that on to the nephrologist.
Not sure about that. Maybe Francesco has an idea.
It's actually rare. What we did is actually we contacted people in Paris that are using alpelisib for PROS, and they're using it at much lower dosages. At these lower doses, actually most patients do fine and have very few side effects. I mean, the side effects that Jenny just mentioned, side effects that are frequently reported in patients treated for cancer, and these patients would get between 2 and 3 times more drug than the ones proposed. Okay.
Matias, you've shown that complement activation rescues your actin phenotype. Did you also have a chance to look at the other phenotypes, like the size of the cells and these additional slit diaphragms?
Yeah, this is a good question. So we're working as we speak on this question, whether the slit diaphragms are rescued in their localization, and this is EM that it is taking unfortunately a little bit. For the cell size, we think there is a also rescue. Yes.
All right, I think there's another question by one.
Yeah, it's a related question, Matias. So I mean, it's always fascinated me that some patients with those mutations present with steroid-resistant nephrotic syndrome. So massive proteinuria, glomerular disease predominantly. So in terms of in your mouse knockout model, and perhaps even in humans when occasionally you get biopsies, have you seen the same changes, for example, in slit diaphragms? Those look very specific to that disease.
I think the glomerulus in the knockout mouse has not been studied so well yet, and we should go further here and also do EM studies on the glomeruli and study the nephrin and podosin molecules just to see if they are maybe misplaced. This is not so easy, obviously. This is very tiny structures, but we definitely want to try that. Yes.
Okay, very interesting stuff. And now we are heading for lunch.
syndrome, temporal talks will be on cystinosis. But
Thank you, Max. Thank you very much. So, what I will do, does this work? But the arrows don't work.
I thought I fixed it. No?
Okay. So, what I will do is that, and I hope you apologize, I will not go into depth of all the new approaches that is taken in cystinosis, simply because I chosen to give you sort of an overview. So I will be sort of superficial for some aspects of the data that I will show you, but it is so that I can show you everything. So this is basically my only introductory slide on cystinosis, and I take it that most of you are familiar with the disease. Suffices to say that in this disease, lysosomal cystine efflux is impaired because the transporter that allows the efflux of cystine from lysosome doesn't work. And as a result of this, cystine accumulates virtually in every tissue and very often also forms crystals and causes a variety of symptoms that involve most organs. The disease starts with the kidneys, with the Fanconi syndrome that progress to end-stage kidney disease, and with crystals in the eyes. But as you can see, all other organs are affected. And we do have a treatment for cystinosis, which is cysteamine. And cysteamine allows the efflux, the indirect efflux of cystine from the lysosome by forming a mixed disulfide that can exit the lysosome through another cationic transporter that is on the membrane of the lysosome. So before telling you about other treatments, one thing that I wanted to mention is that cysteamine probably should be used better. And we now have cohort studies from a very large cohort of patients. And we have a sibling study that very clearly show that starting very early treatment is of paramount importance to prevent early onset of end-stage kidney disease. So what you see here is the age at start of cysteamine and the risk of developing end-stage kidney disease. And you can see this nearly linear relationship. And you see here in this sibling study that the second sibling, especially if it was treated before any symptom, did much better. So before going to other treatment, remember that cysteamine is a good treatment and that starting early is very important. And starting early could also mean doing genetic screenings at birth. And we now have here an experience in southern Germany that was positive by using next generation sequencing. Patients could be diagnosed even at birth. No. Now, I told you that cystinosin is due to a mutation of this transporter. The name is cystinosin, is a cystine proton syn transporter. So the energy comes from the proton ATPase, that is the gradient of protons. And a few months ago, the 3D structure of cystinosin was resolved and was published and actually confirmed everything we knew. So cystine is admitted into the transporter where it binds to its groove, and it's really the proton that changes the conformation of the protein and releases cystine into the cytosol. What is more important is that we also have learned that cystinosin interacts with many other proteins, and these involve the proton ATPase, but also a number of other proteins in the cytosol. And it's probably because of that cysteamine is not sufficient to treat completely the disease. And so there are very different aspects of the disease which have been uncovered over the past, I would say, 10 to 20 years. So in addition to the accumulation of cystine into the lysosome, so a lysosomal storage disease, we now know that trafficking inside these cells is impaired. We know that there is altered autophagy, that the mTOR pathway is probably activated, and that the TFAB transcription regulation is also upregulated in these cells. In addition, remember that cysteine is the limiting amino acid for the synthesis of glutathione in the gamma-glutamine cycle. And these cells actually, they don't really lack glutathione, but they cannot make glutathione if they're exposed to oxidative stress. And that oxidation can cause stress to mitochondria, but mitochondria themselves, if they're damaged and they're not clear from the cytosol, can produce reactive oxygen species. And another type of autophagy, which is impaired chaperone-mediated autophagy, the chaperone-mediated autophagy is also impaired in these cells. So a much more complicated disease than what we thought. And the question that is still unanswered in many of these aspects is what comes first and what is a consequence. So if I were to put all the things that I told you, plus another one that I will address in a moment, inflammation, I've sort of summarized here in my mind the efficacy of cysteamine in treating all these aspects. And you see that cysteamine certainly is active in clearing lysosomal cysteine, probably acts as an antioxidant, has an impact on preventing apoptosis of these cells, but has very little impact on all these other aspects of cystinosis. And this is where new treatments are actually sought to try to address these other aspects of cell physiology in cystinosis. So I will try to address them now one after the other. I sort of classify them, but obviously there are overlaps. So the first one is inflammation. And when you look at a biopsy of a patient with cystinosis, one striking thing is that in this interstitial fibrosis, what you will see is a number of crystals. These are cystine crystals. And these cystine crystals actually are phagocyted by defective macrophages, and that sits there and cause inflammation. And the end result is that you have the Schwann neck deformation with this atubular glomerula that forms because you have these proximal tubular cells that progressively are destroyed. So this slide summarizes the data that has been gathered in terms of inflammation in the past 10 years. And most of this data comes from the laboratory of Giusy Principe in our hospital, which we have collaborated. And what she has very nicely shown is that pathogen and damage-associated molecular patterns, DAMPs and PAMPs, somehow activate this intracellular inflammation receptor, the node-like receptor protein 2, which cause inflammation. And in addition, cystine crystals can cause inflammation by rupturing lysosome, by damaging mitochondria, and by causing potassium efflux. And this activates caspase-1, and caspase-1 then activates inflammation. Just very briefly, you see NLRP2 upregulated in cystinosis proximal tubular cells. It's a human biopsy, and you see here this increased production of IL-6, MCP-1, and IL-8. And you see here these crystals that when they're incubated with cystinotic cells and with proximal tubular cells, in fact, produce IL-1 beta. And again, this can be inhibited by inhibiting ROS production, rupture of lysosome, or by adding potassium. Another piece of information comes from the laboratory of Stephanie Cherki and Corinne Antignac. And what they've shown is that one of the ligands of cystinosin is actually galactin-3. And galactin-3 is a pro-inflammatory molecule. And so lack of binding of galactin-3 in this very nice and elegant paper was shown to induce inflammation. So can we treat patients with anti-inflammatory strategies? Now we have one piece of information that was generated by Robert Mack, California. So what he did was to treat with anakinra mice around 1 year of age, and he could show. So these mice had inflammation in their muscles. The muscle phenotype of cystinosis actually is not very strong in the mouse models that we have, in the rat model that we have, but at least you could see that they had increased production of IL-1 beta and IL-6, and by treating with anakinra these animals, the muscle fiber size was partially restored, as well as the infiltration with fat. The other aspect of the pathophysiology of cystinosis that has been addressed is the one of impaired autophagy and oxidation. And these and a number of laboratories have studied this pathway. I think the one that has done this more thoroughly is the laboratory of Zurich, of Olivier de Wust, of Alessandro Luciani, which is here in the room. And in this very elegant study, what they've shown is that probably impaired autophagy is an upstream event that leaves into the cytosol damaged mitochondria, which are a source of oxygen species, radical oxygen species, which in turn disrupt cell polarity, induce cell death differentiation, and cause cell damage. And so here the rationale would be that if you target the mitochondria with a scavenger molecule, you would improve the phenotype. And in fact, when they treated this animal with this, I think it was a mitotempo in this case, which is a mitochondrial scavenger, clearly radical oxygen species decreased, but cell proliferation was restored to normal level, cell injury was improved, megalin expression was reestablished, and endocytic processes were rescued. And from an histological point of view, Dr. Chevalier in the US had previously shown that if you treat the same mice with another scavenger, this is METOQ, this Schwann neck deformation could be at least in part rescued. And to finish this aspect on autophagy, I'll simply mention that the laboratory of Sergio Katz at the Scrip Institute is working a lot on the chaperone-mediated autophagy and is testing molecule to rescue this defect, and has shown that by rescuing this type of autophagy, you can also improve the phenotype, at least at the cellular level, of these animals. The other subchapter here would be the one that involves TFAB and mTORC1, and obviously here is actually a continuation with the autophagy defect that I just mentioned. So TFAB is a transcription regulator, and it's a really a master regulation of lysosomal I'm missing the word. Anyway, what it does is actually it regulates autophagy. It promotes, when it's activated, lysosomal biogenesis and lysosomal exocytosis. And obviously, this is a very simplified view. But in lysosomal storage diseases, it is typically upregulated and it is transferred to the nuclear as it is in these cystinotic cells. You see that all of the TFAB is actually in the nucleus. And when we express TFAB in these cells, what we could show is that we promoted the exocytosis of lysosomes and that we restored the normal aspects of lysosome and a lysosomal distribution in these cells. So the idea here would be that with cysteamine, what we do is that we allow cystine clearance from the lysosome. Here, what we try to promote is the dumping of lysosome to the extracellular milieu and the biosynthesis of new lysosome. And there is there are a few molecules that can stimulate TFAB. One of these is genistein, which is a flavonoid, which is very enriched in soy. But here we use it at pharmacological doses. So we treated CTNS knockout mice with genistein for 1 year. And what you could see in these mice is that these huge crystals were very much decreased. And you see that there is a major decrease in the number of cystine crystal accumulated. Cystine levels were also decreased. And these Schwann neck deformities that I've shown you, that are shown here indirectly by this atubular dilated glomerula, were very significantly decreased. The overall aspect of lysosome in the mice, in the kidneys of these mice was much rescued, and the creatinine clearance improved. However, we didn't see a clear improved in the Fanconi syndrome. The other drug that is being tested now in cystinosis is actually everolimus. I told you that the mTOR pathway is upregulated. This work is being done by the group of Alan Davidson in New Zealand and Jennifer Hollywood. What they have shown is that treating of iPS cells with everolimus reduces the number of enlarged lysosome, decreases apoptosis, activates autophagy, but has no effect on cell cystine. Right now, cystinotic rats are being treated with everolimus, and we should know the results of these tests in the coming months. So this slide sort of summarizes what I just said. So we have different approaches that target different aspects of the defect, the cell defect in cystinosis. I would just add that another molecule that we're studying in the lab is luteolin. I don't have the time to talk to you about it. And that the laboratory of Minnie Sarwell in the US has also generated some interesting data where targeting the vacuolar ATPase actually improve the phenotype, and here again in vivo assays are being made. And then I will finish by saying a few words on gene therapy. So this is the last approach that is being tested for cystinosis. So I would just mention this initial work from the laboratory of Elena Levchenko. What they have found is that you could recover a lot of undifferentiated cells from the urine of patients with cystinosis, and that these cells express kidney progenitor markers, and they could be differentiated into podocyte and into proximal tubular epithelial cells. And they could be rescued with a lentiviral vector strategy. And so these cells potentially can serve now to be re-injected in these kidneys and in part to repair damaged kidney. Now, obviously, this is a very initial project, but it's one to be followed in the coming years. And then it was already mentioned this morning to you, this other work by the group of Paul Goodyear in Quebec and in that part of Canada. The prevalent mutation is a tryptophan 138 with a stop codon mutation. And so this is a very good mutation, a good candidate for a read-through strategy. And this aminoglycoside was tested, showing that you can re-express CTNS level, although it's not a completely normal CTNS protein, and cystinosin protein, and this study is ongoing. There are some toxicity issues that needs to be resolved, but it's potentially an interesting treatment for this patient that had this specific mutation. And then I will finish with the hematopoietic stem cell transplantation. So this strategy was developed by Stéphanie Cherqui, when she was working in the Scrip Institute. What really happened is that she tried to do bone marrow transplantation in a knockout mouse. And what she observed is that the improvement in cysteine content was really disproportionate to the number of engrafted cells that there was in this kidney, suggesting that these engrafted cells had sort of a paracrine effect and could rescue the neighboring cells. This was the initial observation. And then a lot of work has been done in the past 10 years. And to make a very, very long story short, what was shown is that probably the primary mechanism is that macrophages that are rescued by the bone marrow transplantation migrate into the interstitium, and they make tunneling nanotubules that can transfer material, be that lysosome or mRNA, and that can rescue the neighboring cells. So one first transplantation was actually performed in Louvain by the team, again, of Elena Levchenko. And unfortunately, this was an allogenic hematopoietic stem cell transplantation from an HLA identical donor. And unfortunately, the patient developed a very severe graft versus host disease, graft failure, and eventually passed away after 2 years. But they could already observe that the number of crystals here seemed to have decreased, and a number of transdifferentiation or of rescue of neighboring cells was observed in different tissues. One thing that I would insist is that with this technique, what you clearly restore is the macrophage pool. So that macrophage pool can now clear these cystine crystals, which are in the interstitium. So the FDA, the FDA has approved an initial phase 1-2 trial enrolling 5 patients, and these 5 patients have been transplanted over the past 2 years. In fact, 3 patients had a renal kidney transplant, and the other 2 patients had advanced CKD. And I'll just simply show this slide that Stephanie allowed me to show, showing that for the most part, the vector copy number was satisfactory, and that cysteine levels decreased. And there are now some very initial results showing that cystine crystal depositions seem to be reduced in these patients, but these are very, very preliminary data. And one very peculiar observation, which was sort of unexpected, is that the color of the hair of this first patient changed. Now, this is probably the less important phenotypic effect of bone marrow, but it speaks to what it does. Because you know that melanosomes are actually derived from lysosome. And this is why patients with cystinosis are blonde and they have light hair. So the fact that after bone marrow transplantation, the hair becomes darker and the skin is darker is probably indicative of something that is happening with this treatment. And with this, I'd like to acknowledge the contribution of people in our lab. We mentioned especially Francesco Bellomo, who did the ketogenic diet, the studies, Laura Rega, Esther De Leo for the genestin study, Anna Tarante, who participated basically in most of these studies, collaborators in the hospital and outside the hospital, and particularly the laboratory of Olivier de Verist and Elena Rechenko. And I thank you very much.
Thank you very much indeed for a wonderful overview. Are there questions from the audience? Maybe I can start. I was intrigued by the story of the ketogenic diet, because as you will know, in the PKD field, also this is a huge topic. An alternative approach that is discussed in the PKD field is intermittent fasting or just delivering ketone bodies. I wonder whether there's any data on this for cystinosis topic.
So this is something that we have obviously discussed a lot. So intermittent fasting in an animal facility is extremely difficult to do. It's possible, but it's sort of difficult to make. The problem is that these patients are sick. These patients are usually catabolic, and our challenge, you know this very well, for us is to get these patients to eat. And so we felt that this was not the optimal approach. This was the basic consideration. So, and this is why we're looking into, triethanoline treatment in addition to a modified Atkins diet, because a standard ketogenic diet is actually hypocaloric. So this is certainly something you don't want to do in a patient with cystinosis.
Question in the back.
Yeah, Economic Stockholm, Sweden. Yeah, I would like to ask you about this allogenic stem cell transplantation. Do you think it's a way forward or not? There was just one patient published, and as far as I know, nobody else is doing that.
So actually, there is another patient that has been transplanted, and I think I think I can speak to that because Paul Grimm actually talked about it at a meeting. So at Stanford, there is another patient that has been transplanted. And here, the philosophy, the idea behind that transplantation was to do what they have recently done with the Schimke paper, the Schimke patients. So the idea would be that patient is on advanced kidney failure. And so what they reasoned to do was to do a bone marrow transplantation using the bone marrow from the kidney donor, a haploidentical transplantation with the idea to transplant then the same kidney, the kidney from the same donor, and being able to do a kidney transplant without immunosuppression. So I know that a second transplantation has been performed, I think 3 or 4 months ago, for sure.
Thank you, because we are considering to do that, but I talked to Elena Levchenko about it, but she was very much discouraged because of the severe GVHD disease.
Yeah.
There's one question in the chat. For countries that don't have access to white cell cystine monitoring, can you please elaborate how we can use cytotriiosidase assets?
No, I cannot elaborate on that. As people in the field know, the team of Leuven has shown that ketotriiosidase, which is a marker of macrophage activation. So ideally, if you have the idea is that if you have little cystine crystals in your body, you will not have a lot of macrophage activation, and then ketotriiosidase level will be lower. And this could be a better marker on how you're doing, because cystine levels actually reflect the cysteine that you've been taking the day before, not what you've been taking the past month. So the idea is to use ketotriosidase. Clearly, what they were able to show is that in patients that seem more compliant, the levels were lower, but I think that there needs to be prospective studies that demonstrate this better.
All right, thank you very much, Francesco. And we'll go on to the next talk that is given by